Hello, everyone. Today we're diving into a crash course on patterns of inheritance.
We are. We're going to take this huge, complex world of genetics, from pea plants to human diseases, and really boil it down.
Distill it to the essentials.
Exactly. It's a foundational journey. We're going to start where modern genetics began with Gregor Mendel's foundational laws.
Right.
Then we'll explore the variations that make it all so much more intricate, and then we'll connect those abstract rules to the actual physical behavior of chromosomes inside your cells.
And we'll wrap it all up with some unique patterns like sex-linked traits, which have had this huge impact on human health and even history.
They really have.
I think the most interesting part of starting this story is just realizing how wrong people were for centuries. Before Mendel, what was the big idea? How did people think traits were passed on?
The old popular idea was called the blending hypothesis.
Which on the surface sounds perfectly logical, right? Like you're mixing blue and yellow paint to get green?
Yeah, exactly. The thought was that parental traits would mix permanently,
Yeah.
and once they blended, you just couldn't separate them back out.
But there's a huge fatal flaw in that idea.
A massive one. If you always blended traits, eventually the entire population would just become uniform. Everyone would look the same. And more importantly, it just fails the basic test of traits reappearing.
That's the key. You might cross a pure black lab with a pure chocolate lab, and all the puppies are black.
Okay.
But then later, when those black dogs breed, suddenly chocolate puppies reappear.
And the blending idea has no explanation for that. Once you blend paint, you can't just get the original yellow and blue back.
No way.
And that's exactly why Gregor Mendel's insight was so revolutionary. He came up with what he called the particulate hypothesis.
So not blending.
No. He argued that parents pass on these discrete, heritable units, what we now of course call genes, and these units keep their individual identity generation after generation.
I love that analogy that genes are like cards in a deck. You can shuffle them, deal them out in a million new hands, but, the ace of spades is always the ace of spades.
That's the core insight. Inheritance is a predictable shuffling of distinct factors, not this irreversible blend. And that understanding underpins, well, everything we're going to talk about.
So it allowed him to establish the rules for how those cards get shuffled, which brings us to his first big law.
Law number 1, alleles segregate during reproduction. And this law really explains the inheritance of a single trait.
Okay, so to get this, Mendel gave us 4 foundational concepts. First up, genes have alternative versions, and we call those
Alleles, yeah, that's what accounts for all the variations we see, like the purple alleles versus the white allele for flower color in his peas.
Got it. Second, every organism gets 2 alleles for each trait, one from each parent.
Right. And we have specific terms for that. If you inherit 2 identical alleles, you're homozygous for that trait. If you get 2 different ones, you're heterozygous.
And third is the concept of dominance. This is a big one.
Huge. It just means that if those 2 alleles are different, if you're heterozygous, only one of them is visibly expressed. That's the dominant allele. It determines what you actually see, the phenotype.
So the recessive allele is still there. It's just hiding. It has no noticeable effect.
It's there, but it's silent. And that brings us to the fourth concept, which is the law itself, segregation.
Okay.
When an organism makes its gamete sperm or eggs, those 2 alleles for a trait have to separate or segregate from each other. So each sperm or egg ends up with only one allele for that trait.
So if you cross 2 parents that are heterozygous for purple flowers, let's say a capital P, little p times another capital P, little p, that segregation gives you a really predictable result.
It does. When you map that out in the Punnett square, you can see the outcome clearly. The offspring will have a 1 to 2 to 1 genotypic ratio.
1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive.
Exactly. And because the dominant allele masks the recessive one, that 1-to-1 genotype translates into that classic 3-to-1 phenotypic ratio. 3 purple flowers for every 1 white flower.
And that 3-to-1 ratio is the proof. It shows the factors are separating and keeping their identity.
And if we connect this to what's physically happening in the cell, it's not magic. It happens because those alleles are on the same spot, the locus on a pair of homologous chromosomes.
The matched pair, you get 1 from each parent.
Right. And when those homologous chromosomes physically pull apart during meiosis I, the alleles that are riding on them are also separated into different gametes. The cellular mechanics are literally doing Mendel's math.
That's amazing. So that first law covers a single trait. What happens when we start tracking 2 or more traits at the same time?
That brings us to law number 2. Traits are inherited independently. This is also known as the law of independent assortment.
Okay, and what does that state?
It states that each pair of alleles segregates completely independently of any other pair of alleles during gamete formation.
So, in simple terms, the way you inherit your eye color has absolutely no influence on the way you inherit, say, whether your earlobes are attached or detached.
Precisely. They don't hold hands, they're passed down separately. And the classic way we demonstrate this is with a dihybrid cross.
Where the parents are heterozygous for two different traits.
Yep. Let's go back to our Labrador example. We're crossing two dogs that are double heterozygotes. We're tracking two things: coat, color black or chocolate, and vision normal or blind.
Okay, so if these two genes for color and vision truly sort independently, what should the puppies look like? What's the takeaway?
You get the hallmark 9:3 to 3:1 phenotypic ratio.
The classic.
It's the classic. 9 out of 16 will show both dominant traits, so black with normal vision. 3 will have the first dominant and second recessive black and blind. 3 will have the reverse chocolate and normal, and just 1 will have both recessive traits, chocolate and blind.
So if we actually do that cross and we don't get that 9.3.3.1 ratio, we know immediately something's up. The assumption of independence is wrong.
It's the ultimate benchmark. That ratio is the fingerprint of two genes that are on separate chromosomes, or at least very far apart.
Right. But of course, genetics gets much more complex than just simple dominance. There are exceptions.
Oh, many exceptions. So let's talk about what happens when one allele doesn't fully dominate the other.
First up is incomplete dominance. And this is where we actually do get a kind of blend.
It is a blend, but, and this is the crucial difference, it's not the irreversible blend of the old hypothesis.
Ah, okay.
Incomplete dominance is where the hybrid's phenotype is an intermediate mix. Think of snapdragons. You cross a red flower with a white flower, the hybrid isn't red, it's pink.
So the dominant allele's expression is kind of diluted?
It's diluted. But if you then bred two of those pink flowers together, you could get red and white flowers back in the next generation, which proves it wasn't a permanent blend.
Okay, so that's different from the second variation, codominance.
Very different. In codominance, both alleles are expressed, but they're expressed separately and distinctly. They don't merge at all.
And the classic human example here is?
The ABO blood group system. The alleles for type A blood and type B blood are codominant.
So if you have type AB blood, your blood cells aren't some weird halfway point between A and B.
Not at all. If your genotype is IA-IB, your red blood cells fully express both the A carbohydrate and the B carbohydrate on their surface. They're both there, both functional, both totally distinct. That's codominance.
Now we get into another layer of complexity, this time about scope. So how many traits a single gene can control? Or, the other way around. Let's start with one gene having many traits.
That's called pleiotropy. It's the idea that a single gene can influence multiple, often seemingly unrelated, physical traits.
Like one switch in a factory that controls lights in the office, a conveyor belt in the warehouse, and the air conditioning.
That's a great analogy. And the classic, if tragic, example in humans is sickle cell disease. It's all caused by a single allele that codes for an abnormal hemoglobin protein.
Just one tiny change.
One tiny change in one protein molecule. And it triggers this devastating cascade of effects. That single mutation causes red blood cells to warp into a sickle shape, which then blocks blood flow, which leads to anemia, weakness, chronic pain, fever, and just widespread damage to every vital organ: the heart, the brain, the kidneys.
It's terrifying. It really shows how, in genetics, the smallest error in the blueprint can cause, system-wide failure.
It really does. Now, let's flip that idea on its head and talk about polygenic inheritance.
Okay, so this is many genes affecting just one trait.
Exactly. This applies to so many of the traits we see around us, things like human height or skin color.
Right, things that aren't just on or off.
Correct. These traits are influenced by the cumulative additive effects of two or more genes all working together. It's not just gene A or gene B, it's gene A plus gene B plus gene C, all adding a little bit to the final outcome.
And the result of that is continuous variation, isn't it? Instead of just tall or short, you get this smooth spectrum of possibilities in the population.
A bell-shaped curve. And this is also where environmental factors can start to play a role, because that continuous variation allows for subtle nudges from things like nutrition or sun exposure affecting the final expression.
So, moving beyond peas and into human populations, let's talk about decoding human inheritance. Since we can't do controlled crosses on people, how do geneticists figure this stuff out?
Well, they rely on pedigrees. These are essentially just family trees that track the inheritance of a specific trait across generations.
So they can look at a pedigree and deduce the likely genotypes of individuals and figure out if a trait is dominant or recessive.
Precisely. And we now know that many serious genetic disorders follow these predictable Mendelian patterns. Cystic fibrosis, for instance, is a recessive disorder.
Meaning you need two copies of that defective allele to be affected.
Right. And then you have disorders like Huntington's disease, which is dominant. So you only need one copy of the allele. But the really unsettling part about Huntington's is that it's late appearing. Symptoms often don't show up until middle age.
Which is what allows it to persist, right? It's like a genetic time bomb that slips past reproductive age. Carriers can pass that lethal dominant allele on before they even know they have it.
Which raises all sorts of profound questions about genetic testing. And the technology for that testing is advancing incredibly fast. For couples who might be at risk, we have sophisticated modern tech for fetal testing.
Things like amniocentesis or chorionic villus sampling or CVS.
Yep, they extract fetal cells to get critical information about genetic conditions very early in the pregnancy. CVS can be done earlier, around the 10th to 12th week, and gives you faster results.
But both procedures, they do carry a small risk.
They do. A small but real risk of complications, about a 1% chance of miscarriage or bleeding. And that leads directly into these really complex ethical questions.
For sure.
I mean, knowing about early onset diseases is one thing, but finding out you carry the Huntington's gene decades before symptoms appear, that forces a really deep contemplation of your future, reproductive choices, privacy. The information is powerful, but navigating it is just deeply personal.
Okay, let's bring all this back to the physical world. We've talked about these abstract factors separating and assorting, but how does a cell physically make it happen?
This is the unifying theory. This is where genes and chromosomes finally come together. Mendel's heritable factors are genes, and they're located at specific positions or loci on chromosomes.
So the behavior of chromosomes during meiosis is the physical basis for all of Mendel's laws.
It is. The controlled, predictable movement of chromosomes is what makes it all work.
So how does that explain the law of segregation specifically?
The law of segregation is perfectly explained by what happens during meiosis the first. You have your homologous chromosomes, one from mom, one from dad. They physically separate, they pull apart.
And the alleles on them go along for the ride.
Exactly. So the alleles are automatically partitioned into different gametes. The cell literally segregates the factors for us.
That makes perfect sense. But geneticists pretty quickly found a big problem with law number 2, independent assortment. It doesn't always work.
No, it doesn't. And that's because they discovered genes that travel together. The concept of linkage.
So this is a major exception to Mendel's law.
It is. Linked genes are genes that are located physically close together on the same chromosome.
So instead of being on separate chromosomes, like the coat and vision genes in the Labradors, they're physically attached. They tend to be inherited as a single package.
Right. It's like two specific bulbs on a string of Christmas lights. They both come along when you pass the string down. This was first observed really clearly in the early 1900s by Bateson and Punnett, who were studying sweet peas.
And they were crossing plants that were heterozygous for 2 linked traits, flower color and pollen shape. They were expecting that classic 9.3.3.1 ratio.
They were. But they didn't get it. Not even close.
What did they see?
They saw a wildly disproportionate result. A huge excess of the parental phenotypes, the purple long and the redrowned, and dramatically fewer of the mixed non-parental types than they expected.
And that deviation from the ratio was the proof.
It was overwhelming evidence that the genes for color and shape were physically connected. They were linked.
Okay, but if they're linked, how do we ever get those mixed non-parental types at all? If they always travel together, how do they sometimes get separated?
That is the key. They don't always travel together. The mechanism that breaks that linkage is called crossing over.
Which happens during meiosis.
It does. It's the physical exchange of segments between homologous chromosomes. The chromosome strands literally swap chunks of genetic material.
Creating a hybrid chromosome.
Exactly. And that physical exchange is what creates new combinations of alleles, resulting in those recombinant gametes that Bateson and Punnett couldn't explain. Thomas Hunt Morgan showed this beautifully in his fruit fly experiments.
Right. He was tracking linked genes for body color and wing shape. What did the recombination tell him?
Well, he found that most of the offspring, about 83%, inherited the parental combinations. That proved the genes were linked. But crucially, 17% were recombinants.
And that 17% number?
That 17% recombination frequency was a direct measurable consequence of crossing over, physically breaking the linkage between those genes.
And this is where the genius of his student, Alfred Sturtevant, comes in. He had an idea for how to use that information.
He had a brilliant insight. He figured if crossing over is a random event, then the farther apart two genes are on a chromosome, the more physical space there is between them.
And therefore, the more likely it is that a crossover event will happen somewhere in that space.
Precisely. The distance between the genes is directly proportional to the frequency of their recombination. A higher percentage of recombinants means they are physically farther apart on the chromosome.
So you can use those frequencies to actually build a map.
You can. This insight allows us to create a linkage map, an ordered list of the relative locations of genes along a chromosome. You don't need a powerful microscope. You just need to analyze the offspring ratios.
That's incredible. You're building a physical blueprint of heredity just by looking at statistics.
It's one of the great analytical achievements in biology, truly.
So, to wrap up our journey through these patterns, let's hit one final special case. When inheritance patterns depend on sex, we're talking about sex-linked genes.
We are. These are genes located on the sex chromosomes, which is usually the X chromosome.
And this is important because it creates these really unique inheritance patterns, right?
Yeah.
In the human XY system, where women are XX and men are XY, recessive X-linked traits are way more common in males.
Far more common. And the reason is simple. A male inherits only one X chromosome and he gets it from his mother. So if that X carries a recessive allele, there's no homologous partner, no second X to mask it.
Whereas a female having two X chromosomes needs to inherit two copies of that recessive allele to actually show the trait.
Exactly, which is why historical examples like human hemophilia, the X-linked recessive disorder that prevents blood from clotting, had such a huge impact, especially in royal families.
That story is always so compelling. Hemophilia became infamous in the royal houses of Europe.
Yeah, we can trace the allele all the way back to Queen Victoria. She was a carrier. She passed that allele to her daughter Alexandra, who was also a carrier, and through her, it affected her son, the Tsarevich Alexis of Russia.
And the pedigree just shows a perfect flow.
A perfect flow. Carrier mother to affected son. Because he only had that one X from his mother, that single recessive allele was enough for him to express the disease.
So our journey started with a monk in a garden, just tracking simple pea plant traits, and it's taken us all the way through molecular mechanics and royal family dramas. From an abbey garden to our own DNA, it's a profound story.
It really is. And the study of inheritance, it doesn't just unlock our traits and our health risks. It also gives us these incredible tools for tracing our deep evolutionary past. Take the Y chromosome.
Yeah.
Because it passes nearly intact from father to son, barring some rare mutations, researchers can use it as a genetic marker to trace entire male lineages back through millennia.
And that reveals history we could never have known otherwise. That's how they trace something like 16 million living descendants of Genghis Khan.
All linked by that single, relatively unchanged Y chromosome that he passed down 800 years ago. It's incredible.
It is.
The code of heredity, first cracked by looking at the color of peas, is really the most intimate and intricate story ever told. It's the story of life itself. We've covered segregation, assortment, linkage, sex linkage. But for all we've learned, there's still so much complexity.
Yeah.
I'd encourage you to think about this. The vast majority of our DNA is non-coding. So what other profound stories about our health, our behavior, our ancestry might be locked away in those regions just waiting for new analytical techniques to reveal them? It really suggests that the deepest dives in genetics are still ahead of us.